The topic of discussion of this lecture is a group of bioentities which are referred to as enzymes. What are enzymes? Enzymes are biological catalysts forming an integral part of many of the metabolic pathways that are present in cells. They are responsible for conversions of many chemical entities to other chemical entities which are part of the metabolic pathways. The origin of the name enzyme dates back to almost 1880 when Kuhn first discovered these entities, and because he discovered them in yeast extracts, he called them in yeast, whose literal translation or in Greek is enzyme. Enzymes, in terms of the chemical nature, are generally proteins. In fact, 99% of all known enzymes are proteins in nature, although more recently RNA has also been shown to possess this ability to do biocatalysis. If you look at all the enzymes, the common or trivial nomenclature of enzymes uses the suffix "ase" at the end of a word. For example, some of the famous enzymes which carry this type of suffix are amylase, which is responsible for breakdown of amylose; cellulase, which is responsible for breakdown of cellulose; phosphatase, which removes a phosphate group from a molecule. So generally, the name "ase" at the end of a word denotes an enzyme. Although there are exceptions to this, as for example, enzymes like trypsin and pepsin have the suffix "-. But as I said, this is an exception to the rule. Mostly the common names of enzymes still carry the suffix "ase". There are three features of these groups of molecules which are very important in terms of their functioning. One is that they possess very great catalytic power, or in other words, they enhance the rate of reactions tremendously, and they are known for their speed of catalysis. Enzymes are known to enhance the rates of reactions relative to the uncatalyzed reaction by factors of up to 10 raised to 14. Note that 10 raised to 9 is a billion-fold increase in speed. So 10 raised to 14 is a million billion almost increase in rate of reaction as compared to the uncatalyzed reaction. The second feature of these group of molecules called enzymes is specificity. In other words, they are able to take specific starting materials and convert them to specific products. And in that regard, they are very different from several chemical reactions we might be familiar with, in which case there might be no specificity in terms of the starting materials, or you might form a plethora of products where you try and do a reaction, which are referred to as side reactions. Enzymes generally are devoid of such side reactions. A third feature of enzymes, as they exist in biological systems, are that they are subject to a great degree of regulation. So there are mechanisms in place which allows a biological system to control how the enzyme works and indirectly control the flux of reactions through a metabolic pathway.